The topic under discussion is the sources of drugs. Drugs are obtained from six main sources. These include the plant sources, the animal sources, mineral earth sources, synthetic and semi-synthetic sources, the microbiological sources, and the recombinant DNA technology. The plant or vegetable source. The plant source is the oldest source of drugs. Most of the drugs in ancient times were derived from plants. Almost all parts of the plants were used, which included the leaves, stem, bark, fruits, and roots. Leaves. The leaves of Digitalis purpura are the source of digoxin and digitoxin, which are the cardiac glycosides. The leaves of eucalyptus give oil of eucalyptus, which is an important component of the cough syrup. Tobacco leaves give nicotine, while Atropa belladonna gives atropine. The flowers. Poppy papaver somniferum gives morphine, which is an opioid. Vinca rosea gives vincristine and vinbrastine. Rose gives rose water, which is used as a tonic. The fruits. Senna port gives anthracene, which is a purgative. Calabar beans gives physostigmine, which is a cholinomimetic agent. The seeds. Seeds of Nux vomica give strychnine, which is a CNS stimulant. Castor oil seeds give castor oil, while Calabar beans give physostigmine, which is a cholinomimetic drug. The roots. Apica cruhena root gives emmetine, which is used to induce vomiting, as in accidental poisoning. It also has amoebicidal properties. Rauwolfia serpentina gives reserpine, a hypotensive agent. Reserpine was used for hypotensive treatment in the past. The bark. Cinchona bark gives quinine and quinidine, which are antimalarial drugs. Quinidine also has antiarrhythmic properties. Atropa belladonna gives atropine, which is anticholinergic. Hyoscyamus niger gives hyoscine, which is also anticholinergic. The stem. Chondrodendron tomentosum gives tubercularine, which is a skeletal muscle relaxant used in general anesthesia. The animal source includes the pancreas, which is a source of insulin used for the treatment of diabetes. The urine of pregnant women also gives human chorionic gonadotropin, which is used for the treatment of infertility. Sheep thyroid is a source of thyroxine, which is used for hypertension. Cod liver oil is used as a source of vitamin A and D. The anterior pituitary is a source of pituitary gonadotropins, which are used in the treatment of infertility. The blood of animals is used in preparation of vaccines. Stomach tissue contains pepsin and trypsin, which are the digestive juices used in the treatment of peptic diseases in the past. Nowadays, better drugs have replaced them. The mineral or earth sources include the metallic and non-metallic sources and the miscellaneous sources. In the metallic and non-metallic sources, we have iron, which is used in the treatment of iron deficiency anemia. The mercurial salts are used in syphilis. Zinc is used as zinc supplement. Zinc oxide paste is used in wounds and in eczema. Iodine is an antiseptic. Iodine supplements are also used. Gold salts are used in the treatment of rheumatoid arthritis. The miscellaneous sources include fluorine, which has antiseptic properties. Borax has antiseptic properties as well. Selenium, as selenium sulfide, is used in the dandruff shampoos. Petroleum is used in preparation of liquid paraffin. When the nucleus of the drug from a natural source as well as its chemical structure is altered, we call it synthetic. Examples include amethine and bismuth iodide. When the nucleus of the drug obtained from the natural source is retained but the chemical structure is altered, we call it semi-synthetic. The examples include apomorphine, diacetylmorphine, ethinyl estradiol, hometropine, ampicillin, and methyltestosterone. Most of the drugs used nowadays, such as anti-anxiety drugs and anticonvulsants, are the synthetic forms. The microbiological source includes Penicillium notatum, which is a fungus which gives penicillin. Actinobacteria gives streptomycin, while aminoglycosides such as gentamicin and tobramycin are obtained from streptomyces and micromonosporas. The recombinant DNA technology involves cleavage of the DNA by enzyme restriction endonucleases. The desired gene is coupled to a rapidly replicating DNA, either viral, bacterial, or plasmid. The new genetic combination is inserted into the bacterial cultures, which allow production of vast amount of genetic material. This technique has several advantages. Huge amounts of drugs can be obtained, drugs can be produced in pure form, and it is less antigenic. While on the other hand, the disadvantages include that well-equipped lab is required, highly trained staff is required as well, and it is a complex and a complicated technique.